The HPA axis is a neuroendocrine axis essential to mediating our stress response. For the modern day human, stress is usually chronic and psychosocial in nature. However, our body responds the same way as it would an acute stressor. Glucocorticoids are the final products of this response and follow an inverted U relationship. Small, acute increases have adaptive effects on brain and behavior. However, large, prolonged increases are maladaptive. The HPA axis is regulated by the limbic system, with the amygdala activating it, the hippocampus inhibiting it, and the frontal cortex inhibiting the amygdala. The negative feedback system allows glucocorticoids to travel back and bind to the hippocampus, inhibiting further HPA activation. In this video, we will examine various early life factors that predict HPA axis dysregulation at a later time point, as well as negative health outcomes that can arise because of this.
To explore early life factors that may lead to dysregulation of the HPA axis later on, we first looked at measures of social status. There are objective and subjective measures of status. Socioeconomic status, or SES, is objective and involves factors such as income, education, and employment. Subjective social status, or SSS, however, involves a person's subjective perception of his or her socioeconomic standing. For example, participants are asked where they would place themselves on a social ladder. At the top of the ladder are people who have the highest standing in their community, and at the bottom are those who have the lowest standing. Research has shown that both objective and subjective SES impact salivary cortisol levels. For example, in a cross-sectional study by Alexandra Ursaci, they measured salivary cortisol levels in 82 children from diverse backgrounds. They recorded objective factors like parental income and education levels, as well as subjective factors like parental social status and perceived stress. Additionally, they tested executive functions like working memory, inhibition, and attention set shifting. Results indicated that in young children, lower SES was in fact associated with higher cortisol levels. Interestingly, the data seemed to flip at age 11. Those in low SES households showed lower cortisol levels. A proposed mechanism for this finding is that early overactivation puts wear and tear on the HPA axis, making it less responsive, hence the switch from hyper to hypocortisolism around the time of puberty. As for the cognitive tasks, the researchers found that both objective SES and parental SSS were positively related to the child's executive function scores, providing evidence that both objective and subjective aspects of early life experience are correlates of children's stress levels and executive function.
Another early life factor important to the regulation of the HPA axis is family dynamics. A stress-inducing family environment or history can lead to HPA overactivation in children and influence the ability of the axis to function properly later in life. One crucial family dynamic factor that can affect children's glucocorticoid levels is the quality of care they receive from their parents, even prenatally. For example, a large effect was found in children whose mothers underwent adverse events or psychological stress during pregnancy. Those early impacts on their HPA axis activity can be observed all the way into adulthood. Another research study done by Lupin and her team demonstrated that children of mothers who were clinically depressed during their pregnancies showed altered frontal lobe activity and a risk of heightened HPA activation later in development. A study conducted by Gunner and colleagues also showed that children who were put in after-school care programs had increased HPA axis activity compared to those who went home to their families. Such children who were exposed to poor care for long hours had a much larger risk of behavior problems later on in life. Despite this trend of increased glucocorticoid levels in children who receive low parental care, a study conducted by Jones and colleagues found a contrasting result. Children who were exposed to severe deprivation of care like neglect, physical abuse, and sexual abuse portrayed lower basal levels. One mechanism proposed to explain this outcome is the downregulation of the HPA axis in response to long corticotropin-releasing hormone stimulation during the stressful period. In other words, wear and tear on the stress response system in childhood led to less cortisol production in later years. Although much more research is necessary to understand the underlying processes of varying glucocorticoid levels, it is safe to say that stressful family dynamics is a predictor of HPA dysregulation in adulthood. These early stress factors experienced in childhood and adolescence caused by unsteady household relationships and a lack of support could result in chronic disruptions in the HPA axis, which will lead to long-term changes in both physical and emotional functions. A very early life factor that could affect later brain and behavioral development is premature exposure to elevated levels of synthetic glucocorticoids. Women are often administered glucocorticoid steroids during pregnancy to decrease the risk of preterm delivery. Since the placenta is the passage between maternal and fetal environments, it plays a key role in mediating effects of fetal glucocorticoid exposure on HPA axis development. According to a study done by Braun and his team, this is clinically relevant because early HPA activation could greatly contribute to the fetus developing diseases later on in life. This raises concerns about the safety of glucocorticoid treatments because not only could they have long-term neurodevelopmental effects, but also cause cognitive and behavioral disorders. Despite the current lack of known substitutes to treat early pregnancy complications, better designed dosage treatments individualized to each situation are necessary to ensure safe pregnancy outcomes and later child development. Why is this important? The research explored above demonstrates that HPA regulation goes beyond genetics and that there are specific environment-based physiological and psychological mechanisms that influence it. Later on, we'll explore these effects on the mind and body and examine social support as a possible mediator.
Elevated glucocorticoid levels from childhood to adulthood can have long-lasting impacts on brain structures involved in regulating the HPA axis. Glucocorticoids are essential for normal brain function. They initiate terminal maturation, remodel axons and dendrites, and affect cell survival. Too much or too little, however, can impair brain function. How this exposure to stress impacts the brain depends on the brain areas undergoing development at the time of exposure. Brain areas undergoing developmental changes are the most vulnerable to stress. Therefore, from birth to 2 years of age, the hippocampus is most vulnerable. From birth to late childhood, the amygdala is most susceptible. During adolescence, the frontal cortex increases in volume, and stress can hinder this process. Finally, from adulthood to death, the brain regions that undergo rapid decline due to aging are particularly defenseless, namely the hippocampus and the frontal cortex.
Some of the main known mental health outcomes associated with HPA axis dysregulation are depression, anxiety, and OCD. Patients with major depressive disorder commonly have abnormalities in HPA axis. A 2001 study showed that cortisol levels are significantly higher in patients with major depressive disorder. This elevated cortisol level during depressive episodes can cause permanent structural changes in the brain and the other organs. It has been shown that when CRH is administered to animals, it causes several of the symptoms of depression and anxiety disorders, including reduced eating, decreased sexual behavior, disrupted sleep, alterations in locomotor activity, and abnormal response to novel stimuli. Additionally, when CRH is administered to primates, symptoms of depression, including decreased environmental exploration and increased huddling and lying down behaviors, are observed. Numerous studies have reported elevated cerebrospinal fluid concentrations of CRH in MDD patients. Increased CRH appears to be fairly specific to MDD, as normal levels are reported in schizophrenia, dementia, and mania. HPA activation may be associated with structural changes in components of the axis, such as adrenal gland enlargement in MDD patients. Interestingly, this enlargement in the adrenal gland is reversed after treatment. A 2012 study by Faravelli and colleagues showed that in people with anxiety disorders, a positive correlation was found between high rates of early stressful life events and hyperactivity of the HPA axis. One possible explanation for this finding is that persistent elevations in cortisol levels could tune HPA axis activity to a higher level, resulting in damage to hippocampal glucocorticoid receptors, or even a loss of hippocampal neurons. This reduces the negative feedback of CRH secretion, resulting in higher CRH and cortisol concentrations. Existing data appear to support the mediating effects of the HPA axis between childhood trauma and post-traumatic stress disorder. A large number of studies have reported that the onset of OCD is often preceded by stressful events, like increased responsibility, losses, and trauma such as abuse or combat. Moreover, it is well documented that OCD symptoms increase under stressful situations, and that patients with OCD suffer from daily life stress more than healthy controls, perhaps due to the hyperactivity of the HPA axis. Additionally, structural neuroimaging studies of OCD patients observed a dysfunction in the anterior cingulate gyrus, a structure known to be involved in the regulation of the HPA axis.
Cortisol affects not only the mind but the body as well. Cohen and his colleagues found that elevated cortisol levels can suppress immune function and facilitate central adiposity, a major risk factor for heart disease and diabetes. Another study by Bruce McEwen touched on the close relationship between cortisol and catecholamines and their combined effect on the body. During an acute stressor, the body releases catecholamines like norepinephrine that increase heart rate and blood pressure in preparation for fight or flight. Chronic stress, however, puts wear and tear on the cardiovascular system and can lead to disorders and diseases such as strokes and heart attacks. These results exhibit mechanisms by which cortisol can lead to increases in morbidity and mortality. Can the stress response be mediated by outside factors? Emily Hooker and colleagues hypothesized that perceived social support may serve as a buffer to the stress response. High social support, like a nurturing friend and family environment, can allow people to more easily cope with stressors. What they found was quite significant. When social support was low, subjective SES was strongly related to cortisol, meaning those who said they were at the bottom of the social ladder exhibited higher cortisol after a stressor than those who said they were at the top. However, when social support was high, the difference in cortisol responses completely disappears. Apparently, perceived social support serves as a protective barrier against stress for those with low subjective SES.
To conclude, lower SES and parental SSS, stressful family dynamics, depression during pregnancy, and glucocorticoid administration during pregnancy are all early life factors that predict HPA axis dysregulation. As a result, executive function, depression, anxiety, OCD, and physiological functions such as the immune and cardiovascular systems can negatively be impacted later in life. While some of these early life factors have genetic influence, many risk factors are environmentally based. This gives hope that there is an equal amount of environmental protective factors that counter the risk factors and keep the HPA axis from being dysregulated.